Good afternoon, everyone. I'm Xinting, Field Application Scientist from GenScript European. GenScript is a world-leading biotech company providing life science services and product. With gene synthesis, peptide, protein, antibody, and preclinical drug development service capabilities, we are internationally recognized as a leading biotech company specializing in fundamental life science research and early-phase drug discovery services. So next, it's my pleasure to introduce our speaker, Dr. Liu Pei. Dr. Liu is the Senior Scientist of Antibody Department at GenScript. Today, she wants to introduce how to accelerate COVID-19 antibody development via single B-cell screening. This webinar will take around 40 minutes. You can type in your questions in the chat box, and Dr. Liu will answer them at the end of the webinar. If you didn't get your answers during the Q&A session, we will take them down and email back to you. So without further ado, let's welcome Dr. Liu to start the presentation. Dr. Liu, over to you right now.
Hi everyone, this is Pei. Today I will introduce GenScript's single B-cell screening service for rapid antibody discovery. As we know, 2020 is destined to be an extraordinary year. Coronavirus disease 2019, also we call the COVID-19, the infectious disease was outbreak. It can cause severe acute respiratory syndrome called coronavirus 2. So as of 27 May 2020, more than 5.5 million cases have been reported across 188 countries, resulting more than 355,000 deaths. More than 3 million people have under lockdown. The situation is very urgent. So, the World Health Organization declared the COVID-19 outbreak a public health emergency of international concern on January 30 and a pandemic on March 11. So the local transmission of the disease has occurred in most countries across all 6 WHO regions. However, until now, there are no available vaccines, no specific antiviral treatments for COVID-19. Also, there are no available method to detect the load of virus or neuralizing antibody level in serum. So in the face of the epidemic, we have worked together and fought together. Based on the single B-cell platform, GenScript have developed high affinity rapid monoclonal antibodies. They can be used for evaluate vaccine candidates and measure the drug efficacy, access herd immunity, passport immunity, and contracting tracing. So we got the high quality SARS-CoV-2 mAbs. Here is the outline of our topic. We will go through an overview of rabbit mAbs, then single B-cell platform for rabbit mAbs generation. Most important, I will introduce an important case for SARS-CoV-2 antibody development. Finally, I will also review the GenScript beacon-based antibody generation service. So the first part, why rabbit antibodies so popular? Affinity and specificity are higher than mouse antibodies. Here is the answer. First, the development of the rabbit B-cell repertoire is unique, significantly differs from that of other mammals. It proposes a 3-step process. First, the neonatal B-cell repertoire generated by lymphopoiesis in the fetal liver and momentum switching to bone marrow after birth. Although a potential pool of 200 VH genes are present for heavy chain rearrangement, a majority of these are infrequently or not expressed. So they are 10 to 20 D genes and 4 to 5 GH genes. Then, interestingly, the generation of the primary repertoire is highly gauge dependent. The primary pre-immune B-cell repertoire evolving during the first 2 months after birth in gun and the secondary immune B-cell repertoire generated upon B-cell activation by the immunogen binding. So it's the immune response. This postnatal diversification is an Euro phenomenon and has so far only been observed in rabbits and pigs. There are two main mechanisms: the somatic gene conversion and the somatic hypermutation, called the STC and the SHM. So predominantly used only in chicken and rabbit IgG gene diversification leads to the replacement of larger nucleic acid sequence stretches with DNA fragments from the non-utilized VH genes. So the secondary B-cell repertoire is generated upon the antigen-dependent B-cell stimulation. Again, additional diversity is introduced by STC and SHM in both heavy and light chains. These events further broaden the B-cell repertoire directed against a certain set of antigens associated with a specific pathogen. So, besides the unique immune system, another important aspect is the scarcity of the inbred rabbit strains, while most mouse strains are inbred. So also most strategy to generate mAbs are based on the recovery of B cell from spleen, bone marrow, or blood, which are present in higher quantities in rabbits than in mice due to their overall larger body size. Compared to mice, 50 times more spleen B cell can be recovered from rabbit. In addition, the intra-chain diversified bridge between 1680 and 171 in the CK is not found in human and or mouse light chains. So the rabbit IgG is more stability than mouse IgG. As discussed in the upon section, the rabbit use different mechanisms to genetically generate and diversify their primary and secondary antibody repertoires compared to humans and mice, so effectively creating a complementary set of binders for the difficult antigens. So rabbit mAbs have been used extensively as analytical tools in the biomedical research and especially for immunological techniques such as IHC, ELISA, FACS, and Western blotting. Rabbit PABs have also been utilized as an important tool and for the safety maps and for the safety assessments. So far, 11 rabbit mAbs are FDA approved in the vitro diagnostic tools in the clinic. So the mAbs and mAbs derived antibody therapeutics are currently widely used to treat human disease such as cancer and autoimmune disease. So it can be used for the therapeutics. As previously discussed, for difficult antigens, rabbit mAbs is your best choice. Here is a comparison of an anti-beta-actin monoclonal antibodies produced by rabbit and mice. For the mouse, the anti-beta-actin monoclonal antibody produced by GenScript was visualized by anti-mouse 568 and directly compared with the anti-rabbit FITC visualization. So the co-localization image shows that the rabbit anti-beta-actin has significantly more binding than mice, indicating that the rabbit mAbs anti-beta-actin monoclonal antibody has a higher affinity. So here is the application in the diagnostic. In the vitro diagnostic, a variety of IVD assays such as IHC, ELISA, and radioimmunoassay are based on the principles of antigen-antibody binding. So given the critical role of these assays in patient animal care, it's extremely important that IVD assays are carried out to use the highest sensitivity and affinity mAbs available. So in the view image, visualization of mAbs can be accomplished in numerous means. However, the most common method is through labeling of mAbs with radioactive or fluorescent molecules to help track them in the body and aid in qualifying the results. So here for the anti-idiotype antibodies, as we know, the anti-idiotype antibodies is widely used for the PK kit development. It should be high affinity because the PK assay are used to measure the absorption and excretion rates, distribution and half-life of candidate small molecules or mAb therapeutics. Results from these assays help identify the optimal dose or toxicity risks of a therapeutic molecule. Therefore, they need to be sensitive enough to capture trace amounts of their antibody targets in free, bound, and total forms in the biological fluid samples. For the mAb therapeutics, is the specificity and the sensitivity of an antibody towards its target antigen ensure both safety and toxicity concerns. So highly specific antibodies can avoid off-target binding and hence offer safety. Highly sensitive antibodies require low dosage administration and hence cause a minimum level of toxicity. So for the CAR-T therapy, they can help construct the SCF chain of the chimeric antigen receptor, and they generate sensitive and specific anti-ID maps that help with monitoring the efficacy and of the target of the therapy. So sorry. So for the second part, as we present the rabbit monoclonal is very important. So we establish the single B platform for rabbit mAb generation. We established the single B cell platform for rabbit mAbs this year. Like human and mouse platform, there are three main platforms for rabbit mAbs. First, the feature display. As we know, it was invented by George Smith. He discovered that the monoculture protein of filamentous phage can be modified as its amino terminals to present a peptide sequence without affecting phage infectivity. The advantage of phage display is it can higher screening range, short time, only several weeks, if not immunized the animal. Rabbit mAbs were difficult to generate by hybridoma technology for a number of years. So the feature display can provide a strong incentive for exploring the accessibility of rabbit immune antibody repertoires by feature display. For the hybridoma platform, the hybridoma technology is a method to generate a stable cell line that can constantly secrete defined mAbs. For this purpose, the B cell derived from an immunized animal are fused with a myeloma cell line in the presence of by the electronic instruments. So our hybridoma platform is very mature and high efficient, but the screening time is longer after fusion, and we may miss some B cell repertoire, but for beacon, so for the beacon count, so we introduced the single B platform. So I will give you a detailed introduction for this platform. So this is our new single B platform based on beacon. From the comparison, you can see that the hybridoma platform may miss some B cell repertoire during the fusion, but on the beacon platform, we can get the full B cell repertoire, and the screening time can short from 12 weeks to 1 day. So it's the biggest advantages of the beacon platform is very quick. Here is the general workflow on beacon. So the first important step is the preparation. We need preparing the wetting and reagent, including washing the machine and wetting chip. We also need to preparing import and culture reagents and the assay reagents. Second, we will the most important step is load cells. So single cells will be loaded in each pan. The pan efficiency is 80%. For the 14K chip, we can load about 12K cells. So third most important is the assay step. This perform an internal base best assay. So the polystyrene bead conjugated with the AB anti-IgG anti the FC region. So it can bind in the secreted antibodies. Then we use the second antibody conjugated with a fluorescent labeled. So it can we can screening the positive hits Then finally, we will export the B cell to 96 PCR plates. Besides the best anti-IgG assay, we also can do the antigen best antigen assay in the level of a single B. Then we can also do the multiple antigen assay. Most important, we also can do the cell-based antigen cell-based assay. We can use overexpressed antigen cell line. When the secreted antibody binding with the antigen, the cell will be turned green. We have constructed a GFP protein in these cells. Then we can screening the positive hits by the turn green signal. This is a proposal for single B cell antibody discovery on Beacon. So phase 1, if clients can't provide antigen, we need to design and produce the antigen to immunize the rabbit or mouse. Then we will enrich the plasma B cell from the spleen, bone marrow, and lymph nodes cells. Lymph nodes all enrich the membrane B cell from PBMC. For mouse, we will use the commercial kit, CD130H positive cells enriched kit. And for the rabbit, we can use the antigen enrichment method to enrich more antigen-specific B cells. In the phase 3, we will screening the positive hits on the beacon. On the phase 4 and phase 5, we can get the antibody sequence in 2 weeks. Our new platform is an automated, efficient, and comprehensive platform, so we can accelerate your antibody discovery step. So for the SARS-CoV-2 case study, based on the beacon, here is our target. RBD protein. As we know, the virus use a special service glycoprotein called a spike to connect to the ACE2 and enter the host cell. The most important binding region is the RBD protein. So we develop an anti-RBD antibodies. First, we want to use these antibodies to develop an ELISA kit to detect a virus. Second, they may have the potential use for the therapeutic applications. So based on the single platform, we have generated COVID-19 as protein antibodies. Here is the animation display on the loading cells. So you can see the movie. When we load the cell in the channel and we based on the electronic light, the single B cells will pan in the pan. And here is the assay video. If the single B in this pan can secrete antibodies, the Bs in the channel will be shy. So finally, we got 142 antigen positive hits for this project. We tested several S proteins antibodies from the rabbit. So these antibodies have a higher affinity and also diversity. The EC50 range from pigram to nanogram, and the 8 antibodies have 4 epitopes. These 3 clones is from the hybridoma platform. You can see we test we sequenced 5 clones and got 4 positive clones, but the 3 clones share the same epitope. So the hybridoma, I think the hybridoma diversity is, I think the beacon's diversity is better than the hybridoma platform. And also we have made the mouse antibodies on the hybridoma platform. We tested the affinity by the Biaco machine. So the rabbit antibody is 10 to 100 times higher than the mouse antibody. Here is our results. Also, the rabbit has a very good blocking effect on the ACE2 protein. So this is we doing a blocking assay on the ELISA, use the ELISA method. The rabbit mAbs can block the ACE2 protein. And then we test the rabbit MonoRab by using the seed virus neuralization method. So from the results, you can see that our rabbit mAbs can neuralize the virus and prevent the virus from infecting the human cells. So then we conjugated the antibodies with binding and acquired 8 paired antibodies. We choose one of the best pair. Here we choose this pair to develop the ELISA kit. So the ELISA kit has a higher sensitivity. It's about 20 picogram and millimeter, and it also can detect seed virus. We also use the antibody to test the RBD overexpressing cell lines. You can see that it has the results 19, here is the 97% of cells could be sorted. So the antibody has a good specificity. It also can be used for the WB experiment, so with higher specificity. Here we also use the mouse antiserum as the control. You can see at the same level, the rabbit's signal is strong than the mouse antiserum. Here is a summary of SARS-CoV-2 S protein rabbit mAbs. We can meet the needs of different applications. If you're interested, so welcome, contact us. I want to introduce the more projects we have done. Here is the summary. These projects are based on the Beacon platform. For the different targets, the number of positive hits range from 21 to 298. So it's correlated with the type of the targets. For the transmembrane protein target, the number of positive hits is lower than the normal protein. than the normal protein and the peptides targets. So the different projects have different results. So the final part is our GenScript beacon-based antibody generation service. If your project is time sensitivity, or you want to get more clones with diversity, you can choose our beacon platform. You can start by immunizing the animals. After immunization, we will prepare a single B cell suspension by selecting and isolating animal spleen cells, bone marrow, or lymph nodes. Then we will enrich, and the screening step will be completed within 1 day, so it's very fast. In addition, we can start with prepared B cell from customer, such as B cells from rabbit, ferret, or recovered patient. We can isolate PBMC, then enrich plasma B cell or enrich all the memory B cell. So do the screening and B cell activation. No matter what kind of species materials, if you're interested in our platform, please contact us. Currently, we are recruiting beta tester. If you join, you can have a greater discounts. Why we can make the research easy, because we have many patent technologies to ensure your antibody development success. So we have the optimal antigen design tool. It can generate a superior peptide antigen sequence. Also, we have the Immune Plus reagent technologies. Is a proprietary method to break immune tolerance of highly homologous antigens. Also, we can provide gene immunization service for the difficult antibodies. For example, the transmembrane target. You can choose our gene immunization service. So GenScript offers a wide array of customer antibody service. However, besides our services alone, we can offer some amazing advantages to help make your research easy. In fact, we have generated so many highly qualified antibodies that we were even awarded the Site AB award for the most popular customer antibody supplier of 2018 and 2020. On top of our experience, our services offer a wide array of patented technologies to increase the efficiency of your antibody. We also have the full ALAC and OLAC or ICOOK certified of the host to host of all our animals. So we also have the 24/7 PhD level technical support to act as a consultants on each project. Lastly, but most importantly, we offer milestone-based project management consultation and pricing with every customer project. So, okay, everyone. Well, that brings up to the end of our presentation. And now we would like to move to the Q&A.
Okay, thanks Dr. Liu for the great presentation. And everyone, you can also type in your questions about the presentation in the chat box. So let's move to the Q&A session right now. Dr. Liu, I will read the questions. So the first question, what are the advantages of GenScript single B-cell screening platform?
Hi, Ting, thank you. Yeah, this is a good question. As shown in the slides, there are two advantages for this platform. The first advantage is fast. It could perform functional assays and completed the screening in 24 hours. And secondly, it could retain the B-cell repertoire as much as possible. Yeah, this is my answer.
Okay, thanks for your answers. So the next question, how to guarantee the unique single B cell can be separated into a single room?
Yeah, this is a very professional question. To guarantee the unique single B cell could be separated into a single room, the most important parameter is the cell density. We have optimized the cell density before loading cells on the chip, and the optimal cell density is 5 to 10 and 10 to the power of 6. So the other parameter is the rounds of cell panning. As shown in the movie, we can set the times of the cell panning. Our practical data shows that 3 rounds of cell panning could maximize the percent rate of the single cells. Yeah, this is my answers.
Okay, thanks, Dr. Liu. So the next question, whether the chips of the beacon can be reused multiple times?
In fact, we generally don't recommend to reuse the chips. Repeat use will reduce the conductivity of the chip service, which will affect the loading and export rate. And the parameters during the experiment need to be re-optimized. Project's success rate cannot be guaranteed, so it's better not to reuse the chip.
Okay, thanks, Dr. Liu. So the next question, could you please share some more information for the pseudovirus used for the functional neutralization assay?
It's a very important functional assay to identify the antibody, whether it could work on the real is whether it can neutralize the real virus. So the seed virus could express the S protein on its surface and contains the luciferase gene. We have an ACE2 overexpressed cell line. If the antibody could block the seed virus infect the cell line, the luciferase gene can't expressed in the cell line. We could test the signal of the luciferase to assess whether the antibody have the blocking function.
Okay, thanks, Dr. Liu. So the next question. Here are a few questions from one attending. Let's take it one by one. So the first question is, could you please share a little more detail regarding the pseudovirus used for the neutralization assay?
Yeah, it's about some question with the last yes. As I introduced the pseudovirus could express the S protein on its surface and contains the luciferase gene. Yeah, we can. So, we have an ACE2 overexpressed cell line. So if the antibody could block the virus infect the cell line, the luciferase gene can't expressed in the cell line. So we could test the signal of these luciferase and to check whether the antibody have the blocking function.
So the next question, what kind of virus was it? What makes it relevant for COVID-19 antibody discovery?
It's lentivirus.
Okay, what was the cells used for virus to infect?
We modified the lentivirus and the cell line is the hello cell. Yeah, we transfected this cell and screening a stable cell line to use in this functional assay.
Okay, thanks, Dr. Liu. So the next question, compared with mouse IgG, is rabbit IgG easier to be humanized?
This is a very good question. Now there are two rabbit therapeutic drug have been approved by FDA. The first is the VEGF target. It's a treatment for the wet age-related macular degeneration. So the second is a CGRP blocker for the migraine prevention. So from the discovery of the rabbit antibody generation, there are only two therapeutic drug has approved. So it's not very easier to humanize the rabbit IgG. The first reason is its unique IgG structure. The rabbit light chain have also often contain an additional crossing in the position 80 to form a disulfide bond with the constant region in the kappa light chain. So it's very stable for this rabbit monoclonal antibody. So it's a reason if we want to this is different with the human light chain. So if we want to humanize the IgG, we need to remutant creatine acid. The second reason is we don't have a lot of the experience to do the humanization like the mouse IgG. So we need to communicate. We need to do a lot of case and to know a lot of experience to do this work. But I think for the antibody immunization, I think the step is much similar with the mouse, because for the mouse humanization, there are a lot of methods. For example, complementary determine region grafting, and also the specificity determine residues grafting, or use the feature display and de-immunization. And so I think for the rabbit antibody humanization, there will be a lot of cases could be used in clinical in the future.
Okay, thanks, Dr. Liu. So the next question, I feel rabbit IgG shows much stronger affinity against antigen than mouse IgG and human IgG from your data. How do you think the rabbit IgG potential to bind against the small compounds?
Yeah, as I show in the slides, the rabbits have a strong immunity systems, and they have the advantages of diversity to the difficult target. So it's very appropriate for the small molecular antibody generation. And this also our recommendation if our clients have these requirements, we will recommend them to use the rabbit to generate the antibody. The success rate is higher than the mouse. Yeah.
Okay, thanks, Dr. Liu. So the next question, you mentioned that you tested with RBD expression cells. Is this cell line available for sale?
We have this we have this service. If you want to do this test, we can provide this service to our clients.
Okay, thanks, Dr. Liu. So the next question, what is the size of the colony of B cells that you usually get?
For the size of the cloning B cells, usually the different targets maybe we could got different results. For the average, we could got about more than 100 positive hits in one round of the screening.
Okay, thanks, Dr. Liu. So the next question, Beacon machine guarantees the single cell selection. Can we not use this machine for hybridoma method, also for single clone selection?
Yeah, we have considered this application, and also it's a good idea to do the screening. Yeah, we have tested this experiment. It could be used for the hybridoma, for the single clone selection. It's very useful for this on this machine. This machine, another application is to screening the stable cell line on this machine. It's the same with the hybridoma. It's the same with the hybridoma selection. So it's also the machine is very I think is very useful for a lot of work experiments on this platform. It's much easier and very very quickly than the fax.
Okay, thanks, Dr. Liu. So the next question, how are NK cells used for immunotherapy? How these are different from CAR-T?
It's a very professional question. As I know, for the NK, they also I know for the cell in cell therapy, they also have the NK. It means CAR-T is NKT or the CAR-T. Also for other immune cells also used to for this for the clinical application. So as you know, there for NK is very different with the T cell. So it can kill the target directly. Maybe as I know, there are a lot of companies have to want to develop this cell to use in the clinical, but until now there is no nature production used on the clinical. The main reason is maybe we couldn't reverse or stop the NK function if it will induce other side effects, side infection on the clinical, I think.
Okay, thank you so much, Dr. Liu, for your detailed answers. Due to time constraint, we just end Q&A session here. For the questions that we didn't have time to answer, I will email back all the answers from Dr. Liu to you. Also, we will record this webinar available to all of you for review. Thank you so much, Dr. Liu. And everyone, I hope you find the section useful and beneficial for your future work. And additionally, I want to say on June 23rd, we will hold another free webinar about how to empower protein and antibody expression by our coding optimization tools. If you are interested in this webinar, please follow us in LinkedIn to stay tuned for latest webinar schedule. And thank you to everyone for joining again. And thanks, Dr. Liu, for the great presentation. I will conclude this webinar, and everyone has a nice day. Bye-bye. Goodbye, everyone.
Bye. Thank you, everyone.
Bye-bye, Dr. Liu.